Hello, I'm Pierre Marie Allard, working at the University of Fribourg, and I'm happy to present you the work that we have been doing with people of BiCIKL, which had for objective to improve the exploitation and sharing of existing and new knowledge in the frame of the Digital Botanical Gardens initiative. So first, I would like to give you a little bit of background on this initiative, which is actually a pilot where we use the artificial biodiversity hotspots, which are botanical gardens, for a much wider project, the Earth Metabolome Initiative. This project has a very simple yet ambitious objective. The idea is to profile the metabolic content of all currently known species on the planet. So as you can imagine, this should keep us busy for several time, and it will require a vastly coordinated and global effort. The objective are three main objectives: to advance life science, benefit human society, and protect biodiversity. I will not enter into all the detail, but one very important thing is that we identified semantic web technologies as a fundamental tool in order to organize the very heterogeneous data sets that we will have to handle in this project. And so within this knowledge graph that we want to build, there is one very specific link that I would like to talk to you about, and that is the object of this collaboration with BiCIKL, and it is the following one. So the link between a molecule and a biological organism. We found two ways basically to add these links. So they can come from pre-existing knowledge, past knowledge, and we have worked on a tool, an electronic resource called Lotus, to aggregate this knowledge. I will talk to you about it a bit more later. And then we want to massively add results from experimental metabolomics data sets to further complement this found in link. And so the objective of the collaboration with the BiCIKL infrastructures and experts was to devise tools that should help us to improve past knowledge exploitation and help us to improve future knowledge dissemination. So I'll start with the first part and talk to you a bit more about Lotus. So first, why did we start building this Lotus resource? Well, we had this overview that there is a very good resource to find, let's say, genetic material associated to their producing organism, proteins associated to their producing organism, but the landscape is much more complex when you start to look for small molecule and their producing organism. You have a wide variety of databases. Some are commercial, some are of very limited taxonomical focus. And so what we did is that we gathered all the publicly available database of natural products, and there was a heavy work of harmonization, curation, standardization to collect three objects: biological taxons, chemical structures, and the reference of the work documenting this occurrence. And then this has been shared in another knowledge graph, the Wikidata knowledge base, where basically this allows to benefit from community curation. So we think that we have a first step, let's say, toward a valid resource to document the occurrence of small molecule inside biological organism. However, there are still improvements that can be made. So if we gather a large, we cover a wide span of research on natural products. Well, here I've been depicting some specific example. You see the analytics behind these studies have deeply evolved. However, the way to report them has not evolved that much. And in this last week, this is a PDF published last week in Journal of Natural Products. Well, it's still kind of encrypted within a PDF, which is basically made for humans and hardly accessible by machines. So this is one first thing. And there is a second problem, and it's that there is an editorial bias towards new structures of molecules and bioactive molecules only. So this is also why we launch in parallel this Earth Metabolome Initiative to try to have an unbiased coverage of this metabolome. But let's come back to Lotus. So we know that there is still a lot of knowledge accessible out there. And so this was the objective of the collaboration with the expertise of Plazzi to extract biological organism out of a relevant corpus of publication. So for this, we started with phytochemistry, which is a typical natural products journal. And the team at Plazzi has been extracting the corresponding treatments out of a set of more than 1,000 of these PDF. And so what we get basically is that we pass from this PDF that we have here on the left with just the simple name of the species to a completely new digital object, in this case, the taxonomical treatment, benefiting from a permanent identifier and a wide set of interesting associated metadata, such as the taxonomy, for example, but also the JBIF dataset, so the occurrence, in this case, of this specific taxon, the occurrence of the related species, and all this information is available in linked open data format. So this was for the biological object. And then we have worked with people at CBILS, so the literature services of the Swiss Bioinformatics Institute. So CBILS is basically building a search engine mostly oriented for biomedical knowledge with a set of vocabulary that is extracted from their corpus of publication. So here they used the list of chemical structures coming from Lotus and applied their pipeline to make more than tens of millions of annotation across the overall corpus. And basically, what you get out of this pipeline is the following. So we now have an abstract which is annotated with the name of molecule which have been described in Lotus. So we know that there are improvements to be made. So for now, the resource linked was the following one. There is a more up-to-date resource, which is actually where is the original source of Lotus, which is Wikidata. And this, we are working with them, but we know that this shouldn't be that much of a problem. Another very important thing that we want to work on is that once we have extracted the biological organism and a molecule out of a PDF, well, we still don't have that much idea on how they relate to each other. And actually, a molecule can be biosynthesized by an organism, but it can also be just found in an organism. Think about a pesticide within a bee. It can be toxic or attract a specific organism. So here we want also to build on the expertise of CBILS and Plazzi that they have gained while establishing this biotic interaction to describe the interaction between two species and basically translate this between the interaction between species and molecule. So now I pass to the next stage, which was basically working on ways to improve future knowledge dissemination. So here we collaborated with people at BiCIKL, and people at BiCIKL, what they work on are nano publications. So it's a very small snippets of information, which can be basically encoded in form of a knowledge graph. So what the people of BiCIKL have handed us is basically a template to very easily document the occurrence of a chemical compound within an organism. So here there are drop-down menus, and this is a typical output. So you have here a nano publication documenting the occurrence of a given compound by a defined mass spectrometric experiment in a specific sample. And so this nano publication now benefits also from a permanent identifier and can be used in this case to document the occurrence of this specific chemical compound in parallel to a classical publication. So the nice thing about working in a knowledge graph structure is that while we are doing these efforts, collaborating with people at BiCIKL, and that we will follow, there are other people elsewhere in other places of the world that we might not know at the moment that document the antibiotic activity of this given molecule, that document the fact that this plant is endemic of a given country, that it has a specific conservation status. And it turns out that this antibiotic is and has for target a multi-drug resistant bacteria, which, as you know, can be the cause of super serious public health problem. So this specific path in the knowledge graph is what we call a molecular argument for conservation. So hopefully with this work, we are going to be able to gather more and more of these molecular arguments for conservation and help in describing biodiversity in a better way, and hopefully try to contribute to its conservation. So I would like to thank all the people which we have been meeting during this, collaboration and BiCIKL TNA project. So all the people at Platzi, Donat, Felipe and Guido, Deborah and Patrick at Sibils for the extraction of chemical entities, and Tobias and Philip at Knowledge Pixel. And also all the people here that are part of the DBGI, Earth Metabolome or Lotus Initiative, so Manu, Adriano, Anna, Tarcisio. There are much more in this part, but these are the ones which were involved in this project together with BiCIKL.